All right, I think we're ready to go. So good morning, everyone, and thank you for attending our virtual brunch. Most of you probably have at least talked to me or seen me at conferences before. I'm Jacob Lippincott. I'm the Sales and Support Specialist for Proteome Software. Wanted to start briefly with a question. Some of you may be wondering, why brunch? This is kind of replacing our typical user group meeting that we would have at ASMS. And when we decided to go virtual instead, we realized that since people would be scattered around so many different time zones, it it seemed kind of fitting have a brunch type meeting when you can have coffee, or you can have a mimosa, or you can have whatever, all are acceptable at brunch. So we're trying that this year is just a little thing. So if you're having a cup of coffee or you're winding your day down, we got a good group of speakers lined up, so we're excited to get started. So I think everybody can see my screen with the agenda slide. We got some updates from the development staff. We have a live demo, which is going to be cool. We're going to talk about some ocean proteomics. We're going to close out with a couple talks on DIA. So that should all be pretty cool. I want to do one more slide before we get into it. For people who are looking for some resources, if you have questions about how to use Scaffold, anything about that, check out our website, our help center. Our YouTube channel has a bunch of tutorial videos that you can follow along with. We got some really good documentation. We've been working to kind of build that up over the past couple of years. So check all that out. Of course, if you have questions after looking at our documentation, you can always contact us using sales at or support at proteomesoftware.com. So enough of the boring stuff. Now let's get into our first presenter. So up first, we have Susan Ludwigsen. She's the head of software development for Proteome Software, and she's going to give us a look at the future of the company and how the product line is going to evolve over the coming months and beyond. So let's jump right in. All right, take it away, Susan.
Okay, thank you, Jacob. So I'm really sorry that I'm missing the chance to see all of you this year at our user group and at our booth, but I want to thank you for joining us. And I just wanted to give you a little bit of an overview, a development roadmap of where we're going with our development process. I've been sharing with you over the past few years our evolutionary process we've been on. And this is to give you an update. I think some exciting things are going to be starting to happen over the course of the next year or so. So this is a development roadmap. First, just to review, how did we get to where we are now? Proteome Software got rolling back in about 2005 with Scaffold. And over the years, Scaffold has grown tremendously in complexity and richness. And we've also launched a lot of spin-off products. Oh, whoops, sorry. What's going on here? Okay, to get us to our current position. And we now have 6 separate products. And I know that many of you find it inconvenient to have to use so many separate programs, especially for your DDA analysis. And we are also finding it very difficult to keep improving all of those different products and keep them up to date with new demands and new technologies. So we needed to map out a plan for the future. And our plan is that we will merge the functionality of many of the programs into one, and merge all the products to a single platform to support continuous improvement of all, so that when we add a feature, it can go directly into our various software products. We're already moving down this road, and in just a few months, we hope to make the first really significant step that's visible to all of you when we release new generations of all of our Scaffold products. They'll have a new look and feel, many new features. So let's take a closer look at them. First, we'll be introducing Scaffold 5, which will have searching from raw files for the very first time in Scaffold. Also, it will introduce Percolator as a means of assessing peptide probabilities, support timsTOF through frag pipe output, and it will still contain an option for Q+ and Q+S. Also coming will be a new program, which we're currently calling internally Scaffold Quant. I don't guarantee that these names will be the names that upon release, but that's our internal name for it. This will replace perspectives, and initially it'll provide a full range of quantitative analysis options for label-free data, spectral counting, tick, precursor intensity. It will still depend on Scaffold or other programs to produce MZIDs for input. From Scaffold, you'll be able to directly create files for Scaffold Quant. Scaffold DIA 3.0 will also make its appearance at that time, and it will include Brian's thesaurus program to introduce PTM analysis into your DIA workflows. And our small molecule metabolomics product, Elements, will also be coming out with 3.0, which will have the new experimental design interface and expanded statistical support and other improvements. So that's all coming hopefully by early fall. And then we will continue on with the journey and we'll begin the process of merging our programs. First, TMT and Itraq analysis and eventually SILAC analysis will be merged into Scaffold Quant. And this will replace the need for Scaffold Q+ and Q+S. Along the way, we hope to help you avoid some of the obstacles that I know a lot of you are encountering, such as large file sizes, long processing times, interfacing with other software, and we hope to support some new techniques as we go. So in addition to just our transition, we want to continue improving your experience. So we'll zoom you past the obstacles. And then we'll get to the big step of merging together Scaffold 5 and Scaffold Quant to produce one fully featured DDA proteomic solution, which we're calling, at least for now, Scaffold Pro. And Scaffold Pro will read raw files or search engine results, calculate its own precursor intensities, support labeled quant, PTM localization, and all of the nice flexible experimental design and statistical analysis features that we've been building into our newer programs. So it'll be a full package for DDA proteomics. Finally, that will lead us to our destination. At the end of the road, we'll have a simplified lineup of fully integrated software solutions for DDA, DIA, and small molecules. And as always, we love to hear from our users. So if you have suggestions, feature requests, complaints, feel free to contact me directly at any time. I love to hear input, and we try very hard to respond to our users as quickly as possible. And now, just to let you know that this is all real and it's really coming. I've been promising you that this was all in the works for a couple years, and it's finally coming to the point where you're going to start seeing it. So next up, you're going to see a live demo of the new Scaffold Quant program. And this, what you're going to see, is not actually just the first iteration of it that will be coming by early fall, but a little farther down the road, where you're going to see how it's going to work with Itraq and TMT data. So Caleb Emmons, one of our developers, is going to show you that. It won't have the newest updated look and feel of the GUI, but you'll get to see a lot of really cool functionality. So thank you.
All right. Thank you, Susan. Looks like we have some exciting things coming up. One thing I wanted to mention is that we are doing questions via the question option in the webinar. So if you have one of those, make sure to put the name of the panelist that you'd like to ask directly, and we'll route those questions along. So, all right, next we're going to go to Caleb Emmons, one of the software developers for Proteome software. And as promised, he's going to give a sneak peek and a live demo of Scaffold Quant, our new upcoming program for quantitative proteomics, which is going to be super cool. So I will pass that along to him right now. All right, take it away, Caleb.
Okay, thank you, Jacob and Susan, and I'm glad everybody could make it to our virtual brunch, although it's too bad we can't meet in person. So as they mentioned, I'm going to be giving a demo here of Scaffold Quant, and something we've been working really hard on, and hopefully will soon be coming out so you guys can all enjoy it. So a little outline, I'm going to be talking about what the vision is for it, what types of data we'll support, how you organize your data, how you can use it to analyze protein level changes, both by looking at fold change and by using a statistical test, how you can use the program to visualize your data and its normalization, and then some more advanced options. So let's jump right into it here. Okay, so here's the program, and if it is built on the Scaffold Perspectives platform, so if you're familiar with that, some of this will look familiar. So what I've got open here is the Itraq demo from Scaffold itself, from Scaffold Q+, and I've loaded it as an MZID. So if you can export MZIDs, you can load those into this program. You can also do a direct export from Scaffold Q+ and get your data in that way. So since I've loaded it as an MZID, right now you can see it's in label-free mode, and that just means it's going to act a lot like perspectives. So you'll have spectral counting, you've got different kinds of spectrum counts here. You've got precursor intensity, you've got some non-quantitative display types like percentage of total spectra and percent coverage. And then you've got tick. This is a new feature, so we've put total ion current in for a nice analysis that way, as long as you've got spectra. And then let's go ahead and go up here and choose to enable labeled quant. So right now we've got the options to add Itraq or TMT data in here, but of course we're going to be expanding that and adding SILAC, support for SILAC, and other maybe user-defined isobaric labeling techniques. So this is an Itraq Four-Plex, and you can see here there's a place to put in a purity correction or not, and let's just go with the default, so we'll hit OK. And you'll notice that the one column that we had here has split into the 4 columns of the Itraq Four-Plex. So we've got the 114 through 117 columns. And now the display types available are all related to Itraq. And you can see there's Itraq intensity, log 10 intensity, log 2 fold change, or coefficient of variation. And if we want to go back, we can switch back to label free. This is kind of like switching between Scaffold and Scaffold Q+. You can just do it right there, and it's just all integrated into the same platform here. So we can switch back and forth. And that's great. So this is a pretty simple file. So let me go ahead and open up a slightly more complex one. So this is a TMT 10-plex experiment here. And let me, while that loads, pull up here. So this is from this data set. It's from a paper by Bluebell, Wilmarth et al., and it's from 2017. And the data is freely available from the Proteome Exchange, PXD, and here's the identifier for that if you want to take a look at it yourself. And so this experiment was a two-way analysis where there were 20 mice split into groups of 5 and then respectively fed either a regular chow diet or a high-fat diet. And then they were sacrificed for analysis after either 8 or 18 weeks. And the experiment has 3 MS samples, 3 runs through the Mass Spec, and each of those has 10 TMT channels, so we get 30 what we'll call quant samples. And it's loaded here, so you can see here the 30 samples. Now that's kind of a bit overwhelming, hard to analyze. So the first thing we want to do when we get an experiment like this open is give it some organization. So to do that, switch to the organized view here. And in the interest of time, I've gone ahead and already created the organization for this experiment. So over here, you'll see the 30 quant samples. And then here you'll see the different categories that I've created. I've created a diet category with the regular chow diet and the high fat diet. And I've created a time category with the 8 weeks or the 18 weeks. And then I've assigned those all those attributes to the samples over here. There's various ways to do that. You can drag and drop into folders. You can right click and assign things that way. So it's a pretty flexible interface here to get your data organized the way you want it, assign all the attributes. And then once you've done that, oh, I should mention actually, so you'll see there's, in these 2 folders, there's 10 mice. So each of this is 1 mouse, each of these samples, and then each of these samples is 1 mouse. And then there were, so that only accounts for 20 of the quant samples. And then there were some more used for pooled, for pooled samples. So they mix together all of the other samples and include it, and that's for normalization. Okay, so the next thing is you want to tell the program how you want to view the data. So to do that, you can click here, Configure Experimental Design, and it'll bring up this dialog. And you can see, so we can just walk through this. If you have fractionation, you would indicate that here. And if you have technical replicates, you would check this box. We don't have that in this experiment. So the next thing is to choose a design. So we can have either the basic design, a repeated measures design, or a two-way design. You may think, since there's a time component here, that this is repeated measures, but remember that's when you've got the same subjects that are measured multiple times under different conditions. And in this case, it was actually different groups of mice that were sacrificed at 8 and 18 weeks. So it's not a repeated measures, it's a two-way design. And then we can specify what's the primary analysis category. So we're most interested in diet, so we'll put that here. And our secondary category is time. So we'll put that in there. And then if we hit apply, and switch back to the samples view, lo and behold, you'll see that the 30 columns that we've seen have now gone down to 4. So we've got the chow diet broken out by 8 weeks or 18 weeks, and we've got the high fat diet also broken out by 8 weeks or 18 weeks. And then you can use this control up here to switch. If you want to say switch back to seeing all the samples, we can choose TMT sample here, and we're getting all the columns back. Actually, we're only getting the 20 that are organized in this organization, because remember that the 10 samples weren't part of the actual experiment. They were just normalization samples. So here you'll see the 20 mice samples, and if we switch, we can also roll all the way up to diet, and then we just get 2 columns for regular chow and high fat. So if we switch now back to the time one, so we're seeing all 4 columns here. Now we're ready to start looking at protein level changes. So the first way you might want to think about doing that is looking at log2 fold change. So if I select that here, in this case now the columns are showing the fold change between, so it's the log2 fold change between whatever column you're in and what you've designated as the reference. And here the reference is set to regular chow diet. You could set the reference to more specific things, any specific sample here or any time, diet and time combination, but let's keep it on regular chow diet for now. So this will compare the fold change between, what's in this column and all of the regular chow samples. Now we might want to do a more targeted comparison between just what's in the 18 weeks under high fat and the 8 weeks under high fat and the 8 weeks under regular chow. And so this is another new feature here, which we can do if we bring up this quant dialog and look at the reference samples box here. You can choose use match reference samples. So if I select that and hit apply. Now you'll see what it's done is now it's comparing just the 8 weeks high fat samples to the 8 weeks regular chow samples and the 18 weeks high fat samples to the regular chow 18-week samples. So that's why suddenly these are all 0 because they're being compared to themselves, and these are just being compared to the matching one. So that's a pretty nice feature. Good. So the next way you might want to compare your protein level changes is actually by applying a statistical test and seeing if there's some significance. So let's go ahead and click on this quantitative analysis button again. And the rest of it here is all about statistical tests. So we can choose a two-way Anova for this data, and we can apply Benjamini-Hochberg multiple test correction. And remember, with the two-way Anova, there's actually 3 different p-values you can get. So they're listed here. So you can look at the primary factor effect. Does diet have an effect on group mean? You can look at the secondary factor effect. Does time have an effect on group mean? Or you can look at an interaction effect. Is there an interaction between diet and time? So let's start with the primary factor effect and hit apply. And when you do that, you'll get another column here in the samples view. And in it will be p-values. And these are color-coded. So you've got white means not significant. This orange color means significant at 5%, but not with multiple test correction. And if I sort on these, you'll see sadly in this case that we don't have any that are significant after the multiple test correction. They would be green if they were. But since we've got 1,300 proteins, you end up getting quite a significant multiple test correction. Okay, so and here right here you also got the selector for what kind of p-value you want to look at. So right now it's under primary factor effect, but if I switch here to interaction effect, it'll switch the p-values and they will now be showing the interaction p-values. So if I again sort on them, that'll be giving me the proteins with the most significant interaction. And let's say we want to select one of those and drill into it, get some more information about that specific protein. We can then select it and click on the proteins view and say switch to the statistics tab here. And that will bring up this statistics tab, which shows a table with all of the two-way Anova results, all the sums of squares and all the p-values over here. And then over here, you'll see the interaction chart, which is pretty helpful to see that, yes, indeed, there really, for this protein, was a strong interaction between diet and time. You can see that for the 8 weeks, it went up between the regular chow and high fat, and the 18 weeks one went down between the regular chow and high fat. And while we're here, we can switch to the spectrum tab and we can see that indeed, yep, here are the TMT peaks for this spectrum. So you can get a nice look at your spectra that way. Okay, the next thing that I want to do is look at some of the visualization tools that we have available. So that's under the visualize view here. Oh, actually, I want to make sure that I'm on log 10 intensity here. Okay, so if we switch to visualize, you'll see that we've got a nice volcano plot and we've got a quantitative scatter plot over here. So you can compare different samples that way. And then there's a graph for some protein trends. And we've got, here's a new chart that shows pre and post normalization. So this is the quant samples values before normalization, and they've been brought nicely into alignment here. There's actually multiple steps to normalization, of course, and this is sort of the first one that this graph is showing. And just to talk a little bit about some of the other steps, one of the main other steps of normalization that is currently being used is called IRS normalization, internal reference scaling normalization. And that was a technique developed by Wilmarth and others and actually introduced with this data set. So that's kind of nice. And one way we can visualize normalization is if we switch to the principal component analysis tab here, and if we look at the scores plot, so right now the data is not normalized. So if we look at the scores plot, so you'll see actually 3 really strong clusters happening here, and you might guess what those clusters are. You might guess that they're the MS samples. So let's verify that. One way we could verify that is by editing our summarization here. Bring this dialog back up. And if instead of organizing on diet and time, we switch back to a basic design and put the MS sample or biosample in as the category. If we wait just a second, this should update. And you can see here, it's color-coded very nicely that indeed the 3 clusters that you're seeing are coming from the MS sample. Now that's a bad thing, right? You don't want to have all of your data points clustering based on what MS sample that you happen to put the quant sample in. So, and that's happening because currently the data is unnormalized. So if we switch back to the samples view here and check this little checkbox to turn normalization on, and then switch back to the visualize view and look at the PCA again, you'll see nicely that the normalization has succeeded in removing the dependence on the MS sample, and we've got just some nice overlapping circles. That's what you want in this case to show that the MS samples don't lead to the clustering. Okay, to wrap up here, I wanted to just point out a couple more advanced options that all of this is in progress, so that we're still coding it up, enabling everything, making it all work right. So here's some things we've got coming down the pipeline. For normalization, maybe you've got some internal standard proteins, in which case you want to come to this page and click add protein, and you type in the name of them, and you can select your favorite protein and put it in there. Whatever if you have 5 or 10 or whatever internal standard proteins, you would put them all in there. There's also a box here for designating pooled reference samples. So remember, this experiment did have pooled reference samples, so you would come here and you would choose the attribute, in this case treatment pool, that tells the program which ones are your pooled reference samples, so it can use those. And then we've got another tab here. Again, this is still in progress, being added to, but these are some options to choose which spectra are used for quantitation. Perhaps you want to, by default, we actually would exclude shared peptides, but maybe you want to use them for quantitation, then you can check this checkbox. Got something called reporter ion tick ratio, which looks at the ratio between your reporter ion current and your total ion current, and you can use that to exclude spectra. So, and there'll probably be some more options coming here. So I think those are the main things I wanted to show you today. And yeah, if you have any questions, suggestions, I'd love to hear them. You can contact me directly, caleb@proteumsoftware.com. So thanks for your attention.
All right, thank you, Caleb. Looks like the Scaffold Quant is coming along nicely. All right, let's move to a presentation from one of our guest speakers. Next, we're going to hear from Mac Saito. Mac is a senior scientist in the Marine Chemistry and Geochemistry Department at the Woods Hole Oceanographic Institute. And Mac is presenting a talk entitled Complexity in Ocean Metaproteomics, 20,000 Spectral Counts Under the Sea. So with that, take it over, Mac.
Great. So yeah, thanks for the opportunity to speak today. As mentioned, I'm a scientist studying the oceans, and I went back and looked to see when I first started using Proteome software. It looks like about 12 years ago. This is a picture of one of the field environments we've been working on, Mcmurdo Sound of Antarctica. It's actually a really beautiful place with some whales here, and this is sea ice and Erebus volcano in the background. Let's see if I can change my slide here. So yeah, today I'm going to be talking about how we're trying to apply proteomic technology to the marine environment. And apologies to Jules Verne for this changing his title. But our emphasis has been primarily on wet lab method development and field sample collection to try and bring proteomics as a tool to understand how the oceans are affecting global carbon cycling and how they work both biologically and chemically. And there's a number of major facets of our research. Sort of our bread and butter is microbial proteomics, probably very similar to a lot of the experiments that other people do for model organisms, except we work with a lot of non-model marine organisms, bacteria, archaea, eukaryotic microbes. Because a lot of these microbes are recently in culture or difficult to culture, we're often using custom genomic and transcriptomic data sets for our peptide spectrum mapping. Based on these data sets, then we learn things that we can go out into the oceans and try and understand how natural samples in the oceans work for their proteins. In other words, the metaproteomics of the natural environment with the entire community mixed together in our samples. And a lot of this is global metaproteomic research. That involves custom metagenomic and metatranscriptome data sets for our PSMs. And then we've focused pretty heavily on doing targeted metaproteomics using stable isotope labeled peptides, using multiple reaction monitoring and parallel reaction monitoring. We also do a lot of metalloproteomics. I won't talk about that today, but basically it's a tool that does native 2D separation and then does metal and protein dual mass spectrometry characterization. And there's a recent study here if you're interested in learning more about that. And then this generates a lot of data, far more data than we can use. So we've been building data sharing capabilities, in particular the Ocean Protein Portal, which I'll talk about. And a lot of these pipelines, almost all of them have use Scaffold products, Proteome software products, and we really appreciate that product line because we're so focused on doing the mass spectrometry and the extraction development and the field logistics. Having sort of a really high quality informatics capability that's adapting and growing really has been very useful for us, so we can focus on all the other complicated things we're trying to do as well. And here's just a picture of Woods Hole. If you haven't been here before, it's on Cape Cod, Massachusetts, US. So I often get asked in these ASMS type talks if we go out on the oceans, and so yes, we do go out. Here's a picture of two of the ships that Woods Hole has run over the years. This is the Neil Armstrong and the Nor, which has now been replaced. Yes, we go to sea often from 2 weeks to 70 days all over the world. I've been to every ocean except the Indian Ocean at this point, and we've even managed to bring Brian Searle, the co-founder of Proteome Software, out to sea with us. I would talk to him at meetings, and he would often say how complicated our science was, and after I think many years of badgering him, he finally agreed to come out and take a look and see what we're all about. And so it's been an exciting new collaboration for us. We've just a bit of an aside. We're also developing robots to try and help increase our ability to get samples. The oceans are huge. 70% of the Earth's surface. We're trying to understand how they work and how they're changing. And so collaborating with engineers to go out and get high quality omics samples. This is several hundred liters filtered through a filter here. Using these robots is a big improvement in getting samples back for us. And this is actually one of the cruises that Brian came on with us off of Bermuda. Oops. So yeah, here's just an image of breaking through the ice in Antarctica as well. A lot of these microbes are not very well characterized. This is an example of this, the Southern Ocean. It's very hard to get to. these cruises tend to be 2.5 months long. We're studying a lot of the, if you see some of the brown down below here, that's sea ice algae that are living on the bottom of the ice. So it's an important ecosystem that is changing rapidly as well. Okay, so I'll talk a little bit about our laboratory experiments just to set the context for our field work as well. We often do lab experiments on microbes to try and find new biomarkers for environmental stressors. And this is an example of a study we did a while ago where we took some marine algae, diatoms, shown here, and we starved them for vitamin B12. And these are simply replete on the bottom axis and B12 limiting on the vertical axis, and proteins shown here. And you can see that one protein, this one in purple here, was far more abundant under B12 limiting conditions than without. And this is what actually happens in a lot of our experiments. If you take a key nutrient away from a microbe, it'll have a very strong adaptive response where it, in this case, produce a transporter, produce a protein of unknown function that we then characterize through overexpression to show that it was a transporter. And now that allowed us to have a new biomarker for B12 limitation or B12 stress in an important marine alga in the oceans. You know, one of the things I want to point out is that we're often looking for very big signals, because when you have a unicellular organism and you starve it for something, it will make a ton of a transporter or a chaperone or an enzyme to respond to that deprivation. And so that gives us really nice big proteome targets to go after. And so that's one reason I think this method seems to be working really well in environmental settings. We've also done RNA to protein comparisons. We often hear how RNA and protein don't correlate, but we actually find that they do correlate for some of these environmental biomarkers or stressor proteins because the expression level is so high. So the RNA and the protein kicks up really high, and so you get a very large signal for both if you sample in the right time window. And so with this knowledge of biomarkers that we're gaining from our lab studies, we can go out into the environment and try and see what's happening in the real world. And there's a lot of challenges, though, in trying to do this. One of them is sample collection, getting out there. You need ships. You're trying to get samples from a dilute environment. The analysis is very complicated. The samples are complex with many organisms mixed together. The data analysis is complicated, and of course, then sharing the data is really important because we need to get this out to the community. One of the techniques we've been working with in recent years is 2D active modulation to get really deep into our proteomes. And this is an 8-hour nonlinear 2D gradient that has dual traps and a high pH, low pH set of columns. And it's effectively like 10 or 15 HPLC runs happening from a single injection, but that allow actually very narrow peak widths, so we can get pretty deep into the proteome that way. And that's been working really well for us to get into our samples. You can see this is a 1D versus 2D comparison, and we get roughly 4 or 5-fold more peptide IDs. And here's an example of what some of these biomarkers look like in the ocean. This is actually MRM data using a triple quad, but the targets were built off of discovery using spectral counting. And in this case, we're going from Hawaii to Samoa on this map here, and Hawaii is on the northern end and Samoa is on the southern end, crossing the equator. And you can see nitrogen stress proteins are more abundant in the north and disappear in the south, and the opposite happens for iron. And then we can take these data sets and then interpolate them to make what we call ocean sections. So this is now the north and this is the south. And as we go from north to south, we go from a region of nitrogen stress to a region of iron stress that corresponds to where those nutrients are in the oceans. And so this was really, really exciting to a lot of us because it showed us that these biomarkers really can see changes in how the ecosystems are functioning and how they're responding to their chemical environment. I should point out that there's nothing down deep here because this is hundreds of meters and there's no light down here. So these photosynthetic organisms don't exist down deep. And one of our more recent observations is that as we went down into that dark zone in the ocean, the most abundant protein turned out to be an enzyme that had 23 atoms of iron per complex. So this was a really big shock to us because, as I mentioned before, the upper ocean is iron limited, but then you go down deep and literally the number 1 and number 2 most abundant proteins appear to be iron proteins. And this is consistent with when you take the organism that this comes from and you grow it in the laboratory, indeed, inside the proteomes of those laboratory organisms, those proteins are most abundant as well. And what we think is happening is that this organism is living off of oxidizing nitrate with oxygen, and so it covers its membrane with this enzyme, just waiting for nitrite to come down, and its organic matter degrades from sinking plant growth in the upper water column or algal growth. It's ready to pounce on those chemicals that are coming out of it and create energy, and that's why it's basically covering itself in this. But this is an interesting example. Basically, you know, we're looking at the top 2 most abundant proteins in the proteome and able to write a story about it, and we have you know, 70,000 more to go in terms of describing that story. And one of the things that we've realized is that normalization in metaproteomics is actually fraught with challenges, and it's something that metaproteomics people can debate at no end. But you know, the reason for this is that in the laboratory experiments, we often use normalized spectral counts or normalized spectral abundance factors, or as Gail just talked about, you could normalize the proteins if you're targeting. But in our system, the biological matrix is actually changing with depth, and also there's also the potential for organic matter in the form of non-tryptic peptides from degrading organic matter that's sinking and other organic molecules. And so here's actually the sum of spectral counts as you go down into the water column, and you can see it's really changing quite dynamically. And if we normalized here in blue to roughly 5,000 spectral counts, and then you can see what the profiles of 4 different proteins look like, you can see that the difference between the normalized and the total spectral counts really changes quite a bit depending on where you are in that vertical profile. So one has to be careful about normalizing and question whether or not you want to normalize at all in this context. But the end result is that, you know, for some of these data sets that we're now looking at have roughly 80,000 protein IDs in them, and amazingly, Scaffold's able to handle that as we map to our metatranscriptomic and metagenomic databases. And that basically means that if we have that many IDs, we're getting that many ocean sections, like I talked about before. So here's one section of this more recent expedition we did called Proteomes, and it's a Tmt iron transporter, and it has this really neat distribution pattern right below the sunlit layers of the ocean, and changing as you go farther south. And so we think this is a really valuable record of the ocean's metabolism in space and time. And it could be useful for both basic research and detection of environmental changes, and also for education, for teaching about the ocean or biochemistry or microbiology. And so to share this data, we've been developing a web portal called the Ocean Protein Portal, where you can go in and type your protein of interest. Here's an example of looking for a phosphate transporter, and you can create an ocean section in real time. And this is all currently spectral count data, but it really could be any units that people want to deposit, and it's meant as a resource for the oceans community, you know, for people who are generating data, to share it with people who are interested in enzymes or transporters or microbes, to be able to quickly access more information about a protein of interest. Let's see, so I'm going to go to the next slide. There we go. And one of the key questions when you start working in metaproteomic samples is who is making the protein. And so one of the key ways of thinking about this is to figure out if the tryptic peptide you're studying is unique to a particular taxa or not. And so we have a tool called Metatrip that we just released a version 2 of, that lets you dig down to the taxonomic level of that tryptic peptide. There's a parallel product called Unipept, and the difference between the two is that Metatrip really allows you to have specialized sequencing assemblies, so MAGs and single amplified genomes and metagenome amplified regions. So whereas Unipept is primarily curated model organisms in it, and so that's why we had to create this product. But it allows us to dig down and figure out whose peptide is whose. Let's see, and so more recently we've been exploring using DIA as an alternate means of quantitating this data. And this is in collaboration with Brian Searle here at Proteome Software and Institute for Systems Biology. And it's been pretty exciting so far, where we're taking our 2D outputs, mapping to get our PSMs using our metagenomes, and then creating a list of discovered peptides, and then using PROSIT to generate our spectrum libraries, and there'll be more talks about that in this session later. And then using Scaffold DIA to take our injection data, be it one injection or gas phase fractionation, 3 and 6 phase gas phase fractionation, to generate our peptide and protein reports. In this instance, we've done a 500 to 1100 m/z precursor range with 24 m/z staggered windows for one injection, or 8 and 4 m/z staggered windows for the multiple injections. And here's the performance where we have the single injection actually does pretty well compared to the multiple gas phase ones, but there is some concern of maybe we're getting some peptides in here that are not exactly what we think they are because of small differences in sequence. But so far, it's looking really exciting. We're exploring peptide 1, where the precursor looks really good. Fragments also look really good. So as we would expect, we're getting a really nice DIA analysis of this peptide. But on the second example here, basically we're missing the precursor, but we're getting the fragment ions. So this is actually an instance where DDA would have fallen down and we would have missed this peptide. But now we're actually detecting it. So it's an instance where DIA is giving us more than we would have gotten compared to DDA. And let's see. And so then we can compare what these look like in vertical profiles going down into the ocean. Now here's some of this DIA data. And these are injection triplicates, giving the error bars here. And they look really great. We're getting some real biological structure with more of these enzymes and proteins in the upper water column, where these microbes are more abundant. And it looks similar to our prior MRM data so far. And here's the DIA versus MRM. And I haven't corrected these for the protein in the water column yet. So this is preliminary here for these comparisons, but it's looking like we're getting real signals of ocean proteins and thousands of them simultaneously with DIA. So just to wrap up, I think maybe I should have renamed the title 80,000 DIA fragment ion intensities for ocean protein sections under the sea instead of the original title. And with that, I can wrap up. I want to thank Matt, Don, in particular, our technicians who have done the mass spectrometry and the data and the sample extraction, and the Ocean Protein Portal team, and Brian for collaborating with us. And here's the links for the Ocean Protein Portal if you're interested in using it for research or education. And feel free to reach out to us if you're interested in that, and the MetaTrip capabilities as well. So thanks very much.
Thank you, Mac. That was a super cool presentation, cool science, and the pictures were awesome. All right. Next, we have Nick Vincent-Maloney. Most of you who are attending have probably talked to him recently. He's the head of the sales and support department here at Proteome Software, and he's going to spend a few minutes talking about our new in-house Prosit server. So with that, take it away, Nick.
Thank you very much, Jacob. Hello, everybody. My name is Nick Vincent-Maloney, Manager of Sales and Support at Proteome Software. Thanks again for joining us today. I'm going to talk briefly about Prosit and how we can help you get started using Prosit-derived libraries to improve your DIA searches. Prosit offers high-quality MS2 predicted spectra for any organism on the basis of peptide sequence alone. All you need is a FASTA file. Developed by the Wilhelm and Custer Group at the Technical University of Munich, Prosit is a deep learning algorithm to predict fragmentation and retention times for peptides. We use Prosit to predict DIA-specific libraries from all possible +2 and +3 peptides in FASTA databases. Deploying the source code to a server capable of running deep learning algorithms, for example, a server with a GPU, allows for relatively quick production of predicted spectra for any FASTA database. Then, with a tool like Encyclopedia or Scaffold DIA, you can create searchable libraries for either direct searching of experimental data or generating high-quality reference libraries to use in your DIA searches. Sue and Brian will touch on this in more detail during their presentations. At our Help Center, available at support.proteomesoftware.com, you can find PROSIT-derived libraries for several common organisms, including human, E. coli, coronavirus, Arabidopsis, Drosophila, rat, zebrafish, C. elegans, and mouse. If you would like us to make a PROSIT-derived library for your research, please send us a message at support@proteomesoftware.com. We'll need a FASTA file for your organism of choice to get started. Depending on the size of the FASTA file, we can generate a PROSIT-derived library in a matter of a few hours. So to close out, I'd like to show a sampling of Twitter activity based on the search term PROSIT library. You might recognize some of these Twitter handles. And for more information about using PROSIT libraries in your work, check out our PROSIT page below. For general resources on getting started doing DIA mass spectrometry, see our DIA tips page below. Okay, that wraps it up for me. Thank you very much. Please let us know if you have any questions. We're here to help. And thanks again for joining us, and we're wishing you the best during this trying time.
All right. Thank you, Nick, for that presentation on our new in-house PROSIT server. So if anybody starts to investigate some DIA data, let us know, and we can build you a library, and it's a good workflow. So, all right. Next, we have Brian Searle. Brian is one of the founders of Proteome Software and is currently at the Institute for Systems Biology in Seattle, Washington. And today he is going to present a talk entitled Building Libraries for DIA. So with that, I will pass it over to him.
Hi, Jacob. Thanks again for the introduction. It's too bad that we can't do this in person, but I'm really glad that we can still do some sort of user group style system for you or a presentation for you. The sort of stuff that I wanted to talk about here was kind of more general purpose. How do you actually start to use some of these libraries that Nick was talking about developing and that Mac was talking about using at the end of his session, and how to actually build these libraries and then what they are most beneficial for. So before I begin, as Jacob said, I am a founder of Proteome Software. I also have an a research laboratory at the Institute for Systems Biology, and the funding sources for this are from both sources. So with that, I'd like to talk about library free detection and also sort of the limitations of that style of work. And then second, about building DDA-based libraries and how to best build these libraries and how to best use them. And then second, discussing more about these predicted spectrum libraries, specifically with PROSIT, and how they can be used for DIA and what their benefits and drawbacks are. And then finally close out with some new work on DIA-only libraries. So just to kind of cover some of the basics of DIA again, if you haven't seen this before, I'm just showing a precursor map on the left side, separated by retention time and several precursor MCs. So each of these is a different peptide. Each of these curves is a different peptide. And with DIA, you take a stripe or a section of that precursor mass space and you co-fragment all the peptides simultaneously into the same MS2 spectra. And this gives you fragmentation traces that represent each of those individual peptides. While any of these individual MS2s are often extremely complicated because they have co-fragmented peptides in them simultaneously, if you start to think about time aligning these traces, you can deconvolute some of this information from itself. It's essentially a mechanism to multiplex peptide measurements where you're collecting multiple peptides in the same spectra. So given that, there's a lot of, I think, interest in why DIA is so useful and why people are interested in it. And I think that it gets around a lot of the concerns of doing DDA analysis. And so Max sort of touched on some of these a little bit, but I kind of just wanted to stress what these are and where they come from. So for example, there are many cases in which you'll find situations where you get an early DDA trigger based off of either a shoulder of that peak or another, or actually just another co-eluting analyte. So for example, this is a peptide where there's actually two analytes of exactly the same precursor mass, but only one of them is the peptide of interest. And in this case, you would stick the precursor mass on an exclusion list and never sample it the second time. Here we're able to get really nice chromatographic peaks for each of the different fragment ions for a peptide like this. So this would indicate a type of peptide that would be missing. Another aspect of this is that these precursor traces also can oftentimes be interfered with. So one of the disadvantages of DDA with precursor ion integration is that you really have three different numbers. You've got a precursor, a monoisotopic peak, a +1, a +2 peak. And if any of these are interfered with, well, then that number essentially, or the quantity for that number is in question. And there's not really much you can do to recover from that. Here, if we see interference at the fragment ion level, it's fairly easy for us to say that one of these fragment ions, in this case, there are 10 different or 12 different fragment ions that are possible to be used for quantitation. If one of them is interfered with, then it's very easy to kick that out and use the other 9 to do quantitation with. And then finally, there are many precursors or many peptides that actually don't produce any signal as precursors. And so, for example, this is a peptide where we see really nice fragmentation patterns where they co-elute and indicate the shape of a peptide, but there's actually no signal that indicates that this precursor is present. And this happens actually a lot more frequently than we would think. In fact, all of these three types of things happen a lot more frequently than we think. I took one data set here and then sorted it based on alphabetical sequence and then found all of these threes within the top 30 peptide sequences. So these are all very common situations for us to see peptide fragmentation happening. And it's, I think, worth thinking about why this is going on. So for example, if you were trying to think of analysis in orbit trap, if you were doing normal DDA, you would take a bunch of ions in your precursor scan. You would put all of these ions, pass them through the quadrupole, and then put them into an HCD cell to accumulate so that you would get enough ions to make your measurement. Typically, we accumulate such that you can look at, say, 3E6 ions on a QE or 4E5 ions on a Lumos or a fusion instrument, and then you would measure them in an orbit trap, and then you would see their intensities in the readout. So what happens, for example, if one of those ions, for example, is a little bit lower in population than the others? If the population in the HCD cell is such that most of those precursors are from one or two species, they can actually swamp out all of the signal from any other peptide. And so, for example, that orange trace will just never get seen because it never makes it, or not enough of those ions make it into the HCD cell to fit within that 3-6 limitation. So DIA takes advantage of this, as well as the boxcar method that was developed by the Matthias Mann group. And both of these strategies essentially use the quadrupole to section out peptides so that you're only storing and you're only accumulating certain groups of peptides in the HCD cell. And because of that, you always get some nice measurement. With boxcar, the strategy is essentially just to collect very accurate precursor signals and use that for detection and quantitation. DIA is essentially the same sort of a strategy where you're using the quadrupole as a filter to accumulate different precursor ranges. The only difference is that you get fragment ions that are associated with that peptide, and you can use those fragment ions for quantification. So when you're trying to detect peptides with a spectrum library, for example, or other sort of technique, you'll end up having to extract out a bunch of fragment ions. So this is, for example, a set of signals that have been extracted out from a specific peptide in a DIA file. Where I was interested in trying to measure this peptide. And so it's really difficult to tell which of these signals indicates the actual presence of that peptide. And here I've indicated where it is. It's actually in a relatively low abundant region of the chromatogram. And so here there are two different sets of signals that might indicate the presence of a peptide, or a few different signals. And it's really difficult actually to try to figure out which of these signals is the true signal. So for example, there's that peak in the middle at 64 minutes. There's that peak over to the left at 63 minutes and the one over to the right at 65.5. And it's really difficult to determine which of these peak groups actually represents the peptide. The way we typically do that, so for example, in this case, it's the peak group on the right, is by using these library spectra to be able to interpret the relative abundance of those fragment ions. So as opposed to normal DDA analysis where you would use a search engine that doesn't necessarily know about prior information, here we really need to rely on the presence of prior information to be able to be confident in these types of detections. So for example, if you look at the actual apex of the peak, the mass spec signal there is actually a composite of many different peptides simultaneously. But if you look at the most abundant peaks, you actually see the same fragmentation pattern throughout. And so that is how you derive confidence in this peptide detection. So there are multiple ways of doing peptide detection or peptide analysis here without using anything or any type of library, just using a FASTA database. So for example, this is a strategy called pecan, which is peptide-centric analysis, where you basically take that same fragment ion spectrum or a chromatogram set, and then you score each of those different mass spectrum signals, each of these different time points, you would actually score each of those scans. And at each of those different scans, after deriving a score, you would see that there would be a potential peak in those score distributions. If you were to zoom in around that peak in those score distributions, that would be the way in which you would determine that this peptide was present, and how confident you were in that score set. So for example, Scaffold DIA has a method to do this using a program called Excordia, which essentially is a version of Sequest, or the Sequest scoring engine, but restructured for DIA analysis. So these types of methods can help you detect peptides without ever having seen them before, but they tend to not be as sensitive because they don't know anything about what retention time you should be looking for and what fragmentation patterns you should be looking for. Another way to kind of do this is using a method called DIA Umpire, developed by the Nisvisky lab in 2015, and has been recently re-implemented as direct DIA and Spectronaut and that sort of thing. But these types of methods essentially try to extract out co-eluting fragment ions that are associated with a specific peptide and make a pseudo spectrum. And the way they do that is by looking at a monoisotopic peak and in a precursor signal, and then also look at fragment ions that are associated that seem to co-elute with them. They'll find those co-eluting precursors, map them together, and essentially derive a pseudo spectrum that can be fed into a normal DDA pipeline. These types of methods actually work are starting to work fairly well and seem to be reasonably robust. But there is some disadvantage in them, essentially because they rely on the presence of being able to detect the presence of a monoisotopic precursor peak. As we've seen in the past, many of these monoisotopic precursor peaks don't have the same type of chromatographic traces as the fragment ions that underlie them. And then secondly, many of the times we actually don't even see a precursor peak at all. So libraries really tend to help in these types of frameworks. And so there are many different types of ways of building libraries. And I think the most common way to do this is with DDA. So one strategy for doing that is by taking a DDA experiment or a sample set that you're interested in and adding into it retention time standards. These would be synthetic peptides. They could be, in this case, actual IRT peptides from Biognosys, but they could also be just sort of any type of peptide that you have some sort of a reference for. So for example, the Pierce standard retention time standard is a really good, inexpensive standard that I highly recommend. But you could buy peptides of your own. You could use BSA, digested BSA. You could even actually use peptides that are endogenous in your sample as retention time anchors, as long as you or someone else has been able to map them onto a single line. So the way in which these strategies work is they spike in these specific peptides, in this case in blue, look for them specifically, and then take all of the peptides that they've identified in that DDA run that you don't necessarily know an index retention time for, and then essentially map them on that line based on their actual retention time in this one sample. So this derives what are called index retention times, or IRTs, for each of these different precursors. They're estimates for their retention times, but they're not necessarily, they're a reasonable estimate for what they should be. And the other aspect of this is they can be on a fairly arbitrary scale. So the IRT scale, for example, descends into negative space, negative time, and oftentimes it has much larger values than the actual retention times in minutes in your actual experiment. And so this strategy actually works fairly well if you're doing back-to-back DDA versus DIA runs for building libraries, but it's not very deep, and it also involves a lot of extra work. Another strategy, and so in this case, you would actually take these same IRT peptides and then use your retention times from your first DDA run to actually map onto another sort of DIA run or DDA run. So another strategy actually is to take these types of measurements that you were getting for these IRTs, and actually now that you've known something about these peptides that you've already mapped in a single DDA run, actually try to use those as new types of anchors in the future. And so this is actually a strategy that I was working on with Mac, and we did some of that work that he showed for doing DIA analysis using a DDA library where everything was put onto the same sort of index retention time using an arbitrary retention time rather than a true IRT standard. But the idea here is you would take known peptides that are common to a lot of different samples that are just the equivalent of the BSAs and the actins and the keratins that are frequently found in all of your samples, use those as new IRT samples, and then map them to a series of additional other runs that you could collect in your lab. And so this is a way for developing these sort of mega libraries across many different samples. So for example, Rob Lawrence and I published this paper a couple of years ago where we actually combined over 1,000 different runs, some internal, some external, to develop an entire phospho DDA library that would help us do DIA analysis and PRM analysis in the future. So these are fantastic methods. They do require either stable isotope labeled peptides or using an endogenous peptides inside of your system, but they're a great way to build DDA-based libraries. That said, DDA-based libraries are not without any challenges. So one issue is that they do require a tons of different runs to be able to generate a clear library set. So for example, in all of these cases that we've looked at sample-specific libraries, they really require tens to hundreds of different runs in an experiment. Many times when we try to do these types of experiments, we'll have to look at 40 or 50 different DDA runs just to generate the library itself. And then if you're trying to build a pan-organismal library, you really need hundreds to thousands of runs. The other aspect is that you really also need to use lots of different fractionation methods. In this case, SCX and high pH reverse phase actually provide very different overlaps of types of peptides. Another aspect to this is that different measurements that you would get for fragment ion intensities in DIA actually are somewhat different than with DDA. And the rationale behind this, or the reason behind this, is actually due to different collision energies or different variances in how collision energy is set up for DIA versus DDA. And then finally, the retention times that you get when you've done these types of fractionation methods actually indicate different shifts in patterns and in time because of the fractionation step itself. So in an experiment where you're measuring in an actual matrix, the peptide of interest is actually being monitored in the background of tons of other peptides in general, but when you've done fractionation, you're actually only monitoring that peptide in a background of a handful of peptides. As a result, the retention times for these peptides actually shift and wobble around a bit, as much as 2 to 5 minutes on a 90-minute or 100-minute gradient. So sometimes fairly extensive. So Nick talked a little bit about trying to develop these predicted libraries, and this is actually a really excellent new strategy which has been changing a lot of the ways in which we've been analyzing DIA experiments in just the year that this work has been out. So with a tool like PROSIT, you would end up entering in or giving in a peptide sequence. It would feed that into a neural network and then estimate what it thought fragmentation patterns and retention times would be for that specific peptide. And then these can be used to do library searching in the future. So it essentially forgoes the DDA generation step. I think this is an extremely powerful method. That said, if you search these libraries directly, they still will underperform a little bit. So for example, here I'm showing the number of peptides detected in a yeast run on the left and HeLa on the right of a pecan search, which again, as I said, tends to underperform versus one of these SCX and high pH reverse phase DDA libraries, a combined DDA library, versus just searching a straight predicted library. And the predicted libraries do significantly better than not using a library at all, but they do underperform a little bit versus the DDA libraries. That said, they don't require any DDA experiments. And so you skip the fact that you have to generate this SCX and high pH reverse phase using 16 different injections for the yeast library and 39 injections for this HeLa library. So all of that work is essentially forgone by just using these predicted libraries. So they actually are very powerful, but they're not as powerful as some of these DDA libraries that are built on a sample-specific basis. The reason behind this is actually because of the number of peptides you end up having to search. So here I'm showing you the number of peptides in this HeLa spectrum library is just north of 100,000. It's, I think, 150,000 different peptides sequences. That's significantly less than the number of peptides in the human FASTA database or with different PTMs. And because of that, your multiple hypothesis correction analysis needs to be much more stringent when you're searching all peptides in the human FASTA. And scales as you increase with number of PTMs. This is one of the reasons why we recommend using as curated a database as possible. So for example, UniProt, SwissProt, if possible, and that using this type of strategy, we highly recommend not considering PTMs like oxidation as you would normally do for DDA analysis, or semi-triptychs or unusually high charge states or +1s. In these cases, really searching fewer peptides actually does typically provide you with more detections and better search results. So earlier in this year, we actually published with Lindsay Pino as the lead author on this work. Seth and I, as co-authors, ended up publishing a strategy for developing DIA-only libraries and easily reproducing them, making them very easy to manufacture. And so the idea here is you would take from all of your quantitative samples, so your orange and your blue samples, we would combine them into a pool. And then in this pool, we would acquire multiple gas phase fractionation experiments, in this case 6, just like how Mac was doing for the ocean proteomics, and use these to build a library from, doing one of these fastest searches or using a synthetic library, and then searching your single injection DIA against this gas phase fractionated DIA library. So this strategy develops in-house libraries on your samples, but instead of using 39 SCX and high pH reverse phase, highly difficult to or intensive to acquire experiments, versus just doing 6 gas phase DIA experiments, which are much easier because all you're doing is tuning your instrument to acquire the gas phase fractionation methods. So what is gas phase fractionation? It's essentially just a normal DIA experiment, except it's broken into multiple acquisitions. So for example, if you were acquiring from 400 to 1,000 in a normal DIA experiment, gas phase fractionation only means that you're acquiring in one experiment from 400 to 700, and then a second experiment from 700 to 1,000. This is a strategy that was pioneered with DIA in about 10 years ago in the Goodlet lab, and has been nicely shown in this Sonia Ting paper from 2017 as well for modern methods. For library generation, we typically break this up into not just 2 fractions, but actually 6 fractions. So for example, in this case, we'll monitor from 400 to 500 and 4 MZ wide windows. And after deconvolution, this essentially produces 2 MZ wide DDA quality or PRM quality libraries, except they're based off of DIA methods. And so if you acquire 6 of these, you wouldn't want to do this for every experiment, but it can be really beneficial for doing library generation. So the strategy with this is you would take a FASTA database and you would generate a predicted library using a Prosit tool like the one that Nick provided. And then you would take your 2 MZ wide isolation gas phase fractionated DIA library sample sets, and then you would search these where it's very easy to detect these peptides because they're very easy to see in these experiments. So in this case, we have now situations where we've mapped a predicted spectrum or predicted peptide to a gas phase representation of that peptide. And we can combine all these DIA spectra into a new, what we call empirically corrected chromatogram library or DIA only library. And this can be a nice advantage when you're searching single injection DIA, where you would actually be monitoring this single injection experiment or a single injection experiment set and being able to map peptides that you identified in your library to the single injection experiments. And since everything is acquired on your instrumentation at exactly the same time using DIA methods, it's very easy to line those spectra up and to be able to identify them. So the retention times are extremely accurate now, accurate to about 20 seconds on a 100-minute gradient versus accurate to 5 minutes on a 100-minute gradient. So this becomes a way to use gas phase fractionated DIA methods to refine a predicted library and make it more useful and tuned specifically for your instrument. So this strategy actually works really well no matter what source of library that you use. So even if you use Pecan to build a library to just search a gas phase fractionated data set, you can actually use that new library to detect a lot more peptides than you would typically do. Same thing for the SEX and high pH reverse phase DDA libraries, and same thing for the predicted libraries. That you get a significant boost in the number of detections that you can make. But I think the most important thing is that no matter what the source is, whether you're using building this off of a sample-specific SEX and high pH reverse phase DDA library, or you're building it off of a predicted library, you essentially get about the same results, indicating that they both sort of saturate and provide the top or the highest level of library quality. In this case, being able to do this from just the empirically predicted, empirically corrected predicted library actually only uses 6 DIA runs. So this is a really powerful method for generating libraries with very little effort involved. So how do you build these libraries? I think in Lindsay's paper, this I think is really well spelled out, but I think one of the important messages from this is that you need to monitor when you acquire your gas phase fractionated runs, and this happens because retention times shift over the number of injections on a column until they settle out after about 6 to 10 different injections. And so if you see on this retention time axis on the left, you can see at about 10 injections, you start to see a settling out of the actual retention times. We really need all of the gas phase fractionated samples to have the same retention time axis. And the reason because of this is because they share very few of these peptides. And the aspect here is that multiple or these gas phase fractions typically don't share very many peptides. They only share peptides that are of different charge states. So for example, if a peptide is a +2 in the 600 to 700 range, then it will also be present in the 400 to 467 range as a +3, for example. And you can use this potentially to retention time align between all of these samples, but it's very expensive and very complicated to build this sort of spanning tree to retention time align between all of the gas phase fractions. So we highly recommend just waiting until your column settles out and then collecting them after about 10 runs or so. So given this, where we would recommend starting from is that if you need a really quick answer or you've got a fairly simple matrix, we recommend actually just, DDA actually does really well with this using the normal scaffold pipeline. We think that this is still a really good way to analyze many types of samples if you're not trying to do a lot of quantitation, or you're just trying to get a simple answer for what's in the sample, or you're analyzing gel spots or something that, or gel bands where you have a simplified matrix. DDA performs really well in these circumstances. And so we still highly recommend considering to use it. That said, if you're trying to do a larger quantitative experiment, we find that DIA actually tends to produce better quantitative data in the long term across lots of different samples. If you're looking at less than 6 samples, we recommend this strategy that I believe Sue may talk about in a bit, where you actually use multiple gas phase fractions for single samples, individual samples. So if you've got less than 6 samples, this 2 times gas phase fractionated DIA library, DIA analysis method is actually a fairly good strategy. And here you would just search that with a predicted library. If you have more than 6 samples in your experiment, we recommend this idea about building a gas phase fractionated library and then searching that with one of these predicted libraries for doing that type of analysis. We find this to be a very practical way of generating a very deep library specifically for DIA on your instrument. There are circumstances where you might want to build a DDA library still, and in these cases, might be around PTMs or situations where you have peptides where that are just not very well predicted by the engines like ProZen. So with that, I'd like to just thank some people from Genome Sciences and from Proteome Software that have helped out on this work, as well as Novo and the Prosit group at the Technical University of Munich. So with that, I'd like to close, and I'd be happy to take any questions if there are.
Awesome. Thank you, Brian. That was a great presentation on libraries. Lots to think about, and it's clearly a very important aspect of designing any DIA experiment. So something to spend a fair amount of time thinking about, and we see there's a lot of options to have. Okay, last but definitely not least, we have Sue Weintraub. Sue is at the University of Texas Health Science Center in San Antonio. She is the head of the core facility there, and she is also a dear friend to Proteome software. So I'm going to bring her online. And she is presenting a talk entitled Data Independent Acquisition Mass Spectrometry, Data That's Incredibly Amazing. So with that, take it away, Sue. And
again, thank you all for being here and staying till the end. I'm delighted to be able to talk to you about DIA-MS, which I've subtitled as Data That's Incredibly Amazing. I've been involved in biomedical mass spectrometry. It's hard to believe for me, but almost 50 years, starting when I was 5, of course, and been in proteomics about the last 20 or so. And I consider DIA-MS the most exciting technique that I've used since the introduction of electrospray and MALDI. And that is because it's provided a way of giving comprehensive data for biomedical researchers, but especially in the context of a core where you have limited number of staff. I have 2 people who are amazing, but you really can't do some of the in-depth experiments that others might do. And I'm going to go actually to the end first and say, what are the keys to success for us using DIA-MS in our lab? First, you need to have an appropriate high-performance mass spectrometer. For many years, we've been using an Orbitrap Velos Pro, which was marvelous, but not amenable to some of the comprehensive methodologies for quantitative analysis. You need to have a reproducible, robust sample preparation workflow, which we had that was fine for DDA, but not for DIA at the time. Spectral libraries has been an amazing addition, and especially Prosit, which you've already heard about how much it can do. And then Scaffold DIA. You have to have a way to process the data. In the past, there were possibilities such as through Skyline, but again, in the context of a core, this wasn't always feasible. So when all of these became available, we decided it was time to then move to a much more comprehensive way for protein identification and relative quantification. But then what method should we use? Here I've already given away that it's going to be DIA, but we also had to consider what about reporter ion methods, because they're certainly widely used by many very successfully. We hadn't in the past because the Orbitrap really wasn't suitable for that. And then considering these points below, the reagents are expensive. You need to verify efficiency of derivatization. Prefractionation is really important. Actually, more important to me is the limitations in experimental design and experimental group comparison. For small sample sets, it's fine, but as long as you want to go to larger comparisons, it's much more complicated using reporter ion methods. Precursor ion co-isolation and ratio compression. There are ways you can get around this with instrument scan strategies, but that's at the expense of numbers of proteins you identify and quantify. And then as an analytical chemist, you tend to only have one read per peptide. And so we just decided this was not the time for us to move into reporter ion methods. In the past, the majority of our experiments were done by what we would call gel CMS. And here, make a laser pointer. Each sample would be applied to a 1D gel and separated, and then each gel lane sliced. Each slice reduced and alkylated, digested, run on the mass spectrometer, searched. We use Mascot for that, and then processed in Scaffold, usually using the mud pit option to combine all of the results together. As we were getting ready to start with DIA, I needed a sample to use as a test. And at first we were going to just use the HeLa digest because it's commercially available. But then I realized that any results we get wouldn't be very useful. So I contacted a collaborator, Ratna Vadlamudi, who provided the sample of some control breast cancer cells, cells treated with drug 1, drug 2, and the combo, because they're interested in trying to overcome resistance that happens with certain anti-cancer agents, and often by using a combination, you can have a benefit. And what we did was go through the process now with just one replicate of each and put it into Scaffold 4. And you can see here the total spectrum counts, and blowing it up a little bit, there was 6,025 proteins that were identified. And you can see here that for some selected proteins, clear indication of possible differences in the drug 1, drug 2 compared to either one individually. Especially here, they were very interested. But this is using this cumbersome method. I asked Dana in the lab, how long did it take? Well, she went ahead and outlined every single step from running the gel, processing the gel, excising, extracting the peptides, and that's just for sample prep. When you put it together, sample prep was 4 days, and in Dana's view, this was 4 very painful days of a lot of pipetting. MS analysis was 7 days, Mascot 5, and Scaffold less than 1. Clearly, you can't use this approach for anything very comprehensive and large numbers of samples. And it took 17 days for just one replicate spectral counting. The first step was to work out the sample prep method. And after testing several approaches, we ended up really basing all of our analyses using S-traps from Protify. These are very reproducible and very straightforward to use. So lysis, Protein Quant is very important whenever possible so that you are applying a known quantity to the S-trap. And then also what we have added at this time then, when we moved to DIA, was doing a Peptide Quant. In cases where we wanted to load exactly the same amount of peptide, we knew what it was. And as Mac had said, sometimes you don't want to add the same amount, such as in pull downs or secretome analysis. But taken together, now we had a very robust workflow that was much faster. And for one example, I can show you that for 3 replicates, sample prep was 2 days, MS analysis, 2.25 days, Scaffold, total time 5.25. There were 6,000 proteins, and that included 3 replicates of each of 4 experimental groups. So with this, we felt that we had a workflow and data processing pipeline that we could rely on. I'll show you a few examples. Here, an investigator was interested in chromatin fraction of cells. We put it on the gel first because we weren't sure about sample complexity and how much to load and how much to analyze. And we did our traditional gel CMS first because we were still trying to gain confidence in DIA. This is the more traditional size of 6 slices, again processed the same way. Here it is in Scaffold 4, where there were 2 cells. And in this case, the cell 1 had been transformed in a way to cause then cell 2. 1,700 proteins, 66 with in the chromatin fraction, and there were some interesting results. But then we ran it with DIA, and when I do DIA, I first like to look at the run order. to make sure that there's no systematic error or issues with the instrument. Now notice here, we're up to 3,300 proteins. Consistently, when we compare our gel-CMS to DIA with single injections for DIA, but 6 injections there for the DDA, we get at least double the numbers of proteins or more. Looking very superficially at the results, you can see there's no systematic error. And in fact, you're likely to be able to pick out the experimental groups by the color coding. As Caleb had showed, we can do an Anova here with multiple testing corrections shown in green. And remember that it's very important in DIA to use exclusive peptides. That means a peptide peptide is only assigned to a specific protein, because if you use a shared peptide, you have the risk of having skewed results if you have large differences in the proteins that have shared peptides. So we only use the exclusive, although you're certainly able to look at total as well. And here it is organized similarly to the workflow that Caleb showed with cell 1, cell 2. And if you sort on the p-value, an amazing 2465 proteins were significant with this p cutoff and testing, multiple testing correction. So this was great. What I wanted to look at is what was the PCA, principal component analysis, and not surprisingly, these two separated very well. That's two together in this one spot because there's such strong differences between these cells. Then I wanted to see how well did the DDA data compare and what did DIA give us besides additional proteins. I've shown two specific examples here. Adenine, for example, has no spectral counts in cell 1 and 2 each in the 3 replicates of cell 2. And likewise for this transcription factor, there were clearly more counts in cell 1 than in cell 2. But does blank really mean none? If you look now at the corresponding DI data, you see that in fact there are values. These are log 2, and they are referenced to the middle replicate here. If there were no value, it would say missing value. So there was a measurable quantity, but we have now a realistic representation of the fold change. And likewise for the one that goes down, we find that there are in fact counts in both. So the DIA data gave you more proteins and much more accurate quantitative value. Another fascinating study was looking at some mice that were treated with a vehicle or doses of 2 different doses of drug. And there were 3 different mothers. The offspring were divided into male and female, and then each one received either vehicle, and these were cells, not the mice were treated themselves. The cells from 1 female and 1 male were treated with vehicle, dose 1 or dose 2. After running through DIA-MS, there were 4,800 protein targets, and you can see the PCA. So clearly there's some kind of clustering. Amazingly, they clustered by mother and then by the sex of the offspring. So if the results had been combined, you would never see any of the differences, which the investigator was able to see, both with a dose and a sex and the mother. So here, this was very easy to tell this information, and the investigator now is looking into what is causing these differences. A very recent sample was with some human alveolar lining fluid called ALF that samples were collected from 2 different cities. And the first concern was whether the cities themselves was going to be an influencing factor, because the investigators moved and they had no way of continuing with the samples from the prior city. After processing through DIA, Scaffold DIA, you can see there were 1,700 proteins. And now we can see that the city, so the Ohio samples are green and the Texas samples are blue, they do not clusters separately. This is our pooled sample used for the chromatogram library. Now, it's interesting to see that there do appear to be 2 clusters, and the investigators are looking at this to see what demographic and attribute might be responsible. I'm always concerned that there could be a instrument problem, and I first did the run order and there was nothing there. So in the analysis view in Scaffold DIA, you can look at the alignment of your peptides. Now, this was 22 samples. They were run consecutively with the chromatogram library after about the first 6 or 8. And you can see that after alignment, the chromatography was just fine. This is a very low deviation among them, well less than 1%. So now it's up to the investigator to figure out what are those 2 clusters. Brian had mentioned the idea of using gas phase fractionation for actual samples and not just for library building, and we were astounded to see what happens. When we first started with doing DIA-MS, we were using 12 M/Z windows with 1 gas phase fraction. And this is using some ovarian cancer cells with a knockout of KDM1A. So this was 1 replicate of a control and 2 of the knockouts. With gas phase fractionation for total proteins ID'd, you can see from 12 MZ, you get an increase up to when you use 8 MZ, because we found with our instrument and chromatography, 8 seemed to be just right for the single injection. But if you do 2 gas phase fractions, you get a substantial increase in the number of IDs. But what's even more important is if you look down at the bottom here, is the number that are quantified. Using the 2 gas phase fractions, we only had less than 0.1% not quantified, compared to 600 here in the 12 M/Z windows and 280, 290 with the 8 M/Z. So this was a really big eye-opener for me of ways to enhance your results. What do you need for running DIA-MS? First, you have to have a FASTA library, as you've heard about, that's appropriate for your samples. We've now only used the predicted spectral libraries for either generating our chromatogram library or searching directly. And of course, you need Scaffold DIA. What you don't need is any custom library that you acquire for each experiment. The analysis strategies in my lab, if it's a small experiment, we are recommending doing 2 injections each with 4 MZ windows and searching the ProCit library, as Brian had described. Now, part of the consideration here is the investigator's budget, because we have to charge per injection. If it's a medium size, which is I call 6 to 12, if the investigator can afford it, we still recommend doing 2 injections using 2 by 4 MZ, the gas phase fractions. And on my system, the chromatography is stable enough for this many samples, and the results have been really wonderful. Now, once you start getting much higher numbers, back up and say for 6 to 12, if the investigator chooses, we'll do the single injection and then generate the chromatogram library. In general, this is going to depend on their budget. And then once you get to the larger sets, you're going to do single injections, make the chromatogram library using a ProCit to search. And for our chromatogram library, thanks to Brian helping me look at the data, we're able to use 3 injections of 4 MZ windows and get excellent results. And just so you'll see here, the quantities injected, if we have it, we'll use at least a microgram. And we're fine with 2 micrograms. I've been packing the columns in my lab for 20 years. I even have the same bottle of packing material. It's not ultra thin peaks, but I think it works very well. This is the same gradient that we've been using for many years, and the chromatogram library is a pool. Brian had mentioned there's still times to use DDA instead of DIA. We often get a question, what is the protein in a gel band? That's going to be DDA. Individual samples needing deep coverage, unexpected PTMs, processing sites. We often do this for bacteriophage studies. And I had originally put on here pilot study feasibility testing, but I add now maybe, maybe not, because with the ProCit libraries and gas phase fractionation, unless you have any of the other concerns listed above, I go to DIA and get a bigger number of proteins. The conclusions, as I think you'll agree, flexible experimental design, no need for fractionation. You can have a single MS acquisition per sample. Now that's with having a chromatogram library generated. Clearly enhanced accuracy and precision compared to DDA-based methods for quantitative measures. No need for spike-in standards or custom spectral library. Reproducible workflow, and you can get thousands of proteins, and we do this all the time. And I've got to say that the investigators, I was presenting someone data yesterday, and he truly was amazed and agreed with me of my name of data. That's incredibly amazing. I have to acknowledge the wonderful work of Dana Moeller and Sam Pardo, who have been with me a number of years. UT Health Science Center, the Cancer Center, NIH, and also the University of Texas System Proteomics Core Network for providing the funds for our LUMOS. Kevin Hockala, who worked many years to get the lab going, and now is a thermo service engineer, so he knows how to help us. And I thank you very much, and let me know if you need help getting started with DIA-MS. I really mean that. You're welcome to contact me directly. I'll be able to tell you how we did it and even help with your data and anything to get you going, because I think you will agree it's absolutely amazing.
All right, thank you, Sue. That was an excellent talk. Well, that about wraps it up. I'm going to bring everybody else back online. I enjoyed that we had that kind of three-suite there of talking about DIA and talking about libraries, and then you can see some of the great data that you can present with it. So let's bring everybody back. Let's unmute everyone. Getting there. Make me the presenter. Excellent. Well, thanks everyone. That about wraps up our virtual brunch. I hope everybody got some good science. Thanks to all of our presenters. Sue, Mac, Brian, everybody, Caleb, Susan, everybody in the Proteome software family, Nick too, I don't want to forget about him. Any questions that we didn't get to, we'll answer offline. I thank everybody for joining. I'm going to give a goodbye toast with my mock mimosa this morning. And we hope to see you all in person actually next time. And everybody have a good rest of your day, and we'll be seeing you all soon, hopefully.